Thank you so much for joining us today. My name is Max Sellman. I'm a senior product manager at Aldebaran, covering our portfolio of gene editing nucleases. Really excited to speak with you today about the last decade of CRISPR innovation, really how we've seen from a CDMO's perspective this field change, how the requirements have changed of CDMO partners, and from our perspective, where we see this field going and what's going to be needed in order to, over the next decade, really dramatically expand the reach and breadth of CRISPR therapies. Just a quick disclaimer, products and services at Aldebaran, unless otherwise indicated or for research use only, please contact us if you have any questions about this. Now, you may be familiar with Aldebaran, but if you're not, the real take home about us is that we're a full service CDMO that serves clients across a variety of therapeutic modalities. So whether you're doing cell and gene therapy, making gene edited cells, developing an mRNA therapeutics or vaccines, Aldebaran has the tools, service partnership that clients are looking for to really accelerate those programs. We're proud to be part of Danaher Corporation. So in addition to the power of Aldebaran, we're able to bring to bear through partnership with our sister operating companies, a much broader portfolio. And some other Danaher companies that we would work closely with include, they're not limited to, IDT, Beckman Coulter, Cytiva, SciEx, really a broad breadth of life science innovation companies that we're proud to be a part of. At Aldebaran, when we think about how we're serving these markets with these modalities, really it comes down to phase appropriate research tools to help you translate your therapeutics. So on the gene therapy or gene modified cell therapy side, that means high quality plasmid DNA from research grade through commercial scale GMP. When we're talking about in vivo therapeutics, Aldebaran's a leader in manufacturing mRNA drug substance and drug product from sequence to vial. Finally, near and dear to my heart, clients working in the ex vivo gene edited cell therapy space have really found a lot of success using our off-the-shelf GMP CRISPR nucleases, GMP ribonucleoprotein complexing services, as well as custom protein manufacturing development. Really the goal at Aldebaran is to be the supplier of choice for these tools that you're using to manufacture your genetic medicines. What I wanted to focus on in this conversation, we're really at a pivotal moment in the history of CRISPR and using CRISPR as a medicine. Sometimes it's easy to lose sight of the fact that it's really only been a little more than a decade that CRISPR has been available as a tool for researchers, as a tool for drug developers, in the context of the history of genetic medicine. Of course, this dates back decades. There's been a lot of learnings over the years. But what I'd really like to focus on is the CRISPR era. And I've kind of broken this up into snapshots in time, what we'd say different periods in time in this journey of CRISPR, where Aldebaran has observed some particular shifts in what the field is doing, what the field requires, and talk a little about how we've adapted to serve these challenges. So in the earliest days of CRISPR, to put you in the mindset, the year's 2012, Call Me Maybe is the song of the summer. And the first CRISPR publications are coming out, right? From Berkeley, from Broad Institute, describing this tool that has gone on to really change the world. It was really, I would characterize this time, the first few years, as just the Wild West where there was rapid growth. I think folks who were working with CRISPR at the time recall that tools were hard to come by. A lot of people were home brewing their CRISPR solutions, right? Purifying CRISPR proteins, doing in vitro transcription of guide RNAs. And I think there was a recognition at this point that really what the field needed was just any sort of standard solutions as it worked, towards growing up. This really kind of came to bear in the following era, what I would say, the several years immediately preceding COVID, as CRISPR as a medicine and as a medical field really started to mature, we started to see the first IND approvals for CRISPR medications and the first patients treated. And we think about this is really rapid, right? Just a few years after the initial publications around this tool, we're already treating patients, getting data readouts, seeing a Nobel Prize be awarded around this technology, just a really rapid innovation. And keeping in mind, it hasn't been a smooth, perfect journey, right? I think a lot of folks in this field remember the CRISPR babies controversy. And from a personal perspective, I was really proud to see and be part of a field where there's a very uniform negative reaction to that situation and a really fast alignment within the field that, hey, we're not ready to be doing, germline editing, that much more work is required on this technology. We're just getting into clinical studies. It's a time of a lot of change in the CRISPR space, but a lot of excitement. And, on the Aldevron side, what we were seeing was a need from industry, from academia developing these things to standardize. So as we're moving towards the clinic, there's a desire to get your hands on tools that would enable that rapid access and function, not having to mess around with figuring out if your reagents are going to work as you move into clinical study. So some of the ways that Aldevron approached this, we've been talking about this for years at this point. One of the first things we did was launch research grade and then off the shelf GMP CRISPR nucleases. And the idea here, which made a lot of sense as clinical trials were just getting started, still makes sense today, was that it would enable clients to have access to a much higher quality grade of CRISPR nuclease than was available in the market at a much faster turnaround time and not requiring custom production in order to get there. And this really took off. We saw this get widely adopted as clients realized that this was just a very straightforward way to get access to therapeutic grade CRISPR reagents. Fast forward to today, this really has been proven as a strong strategy. We have upwards of 14 clinical programs that have applied this product, and we now provide regulatory support to our clients worldwide, including having a drug master file here in the US for these for the nuclease, SP-Cas9 nuclease products that we have. Another core innovation at this time, right? The field was realizing and really dialed into the concern about off-target genome editing, right? Everyone, I think, is familiar that there's the prospect with CRISPR proteins that in addition to editing at the target of interest, that there's some low level of off-target editing that in the context of a therapeutic could be potentially very hazardous. And so the field in this era was really focused on how do we minimize off targets? How do we ensure that we're making safe edits? And Aldevron had partnered with our sister company, IDT. At the time, neither of us were part of Danaher yet, but we were already partnered because we realized that the discovery of a high fidelity nuclease that greatly reduces off-target edits would have great therapeutic value. And so Aldevron partnered with IDT to launch this as an off-the-shelf GMP product. And I'm proud to say that you look around the market today, there's a number of tools out there for reduction of off-target effects. This is really the original high-performing, high-fidelity nuclease, and it still stands up today against the other technologies that are out there. It does a fantastic job at its stated purpose of really, really dramatically reducing off-target edits. So in time at Aldevron, this is sort of expanded into a general philosophy for us, right? Which is that we serve clients on a custom basis, of course, but standard processes often require standard solutions. And so today you work with Aldevron. We have a wide suite of off-the-shelf offerings, research and GMP, not just on the nuclease side, but also in vitro transcription and mRNA manufacturing enzymes, standard plasmids for vector production, cloning, reporters. We really run the span here of different standard offerings that clients would need at high quality with kind of associated regulatory documentation. Obviously, COVID was highly disruptive in our industry, and it when that really happened relative to therapeutic developments in the CRISPR space, actually makes for a really nice or really clear bridge point into this, what I would say is kind of our current era, which I'd categorize as a maturing clinical space where drug developers are pushing to get into and through the clinic as soon as possible. So we saw some really dramatic change just over the last few years as some of the next generation CRISPR tools have actually pushed into the clinic. And we've even started to see data come out where not just CRISPR, Cas9 based medicines, but medicines based on base editing technology and prime editing technology have also entered the clinic and are looking really promising. In this period of time, the industry actually got guidance for the first time. Up until this point, we'd really been relying on previous guidances for gene therapy and unmodified cell therapies. And at Aldebaran, certainly, we observed a lot of confusion and uncertainty from our clients about what would be sufficient information from a filing perspective, what quality of reagents would be sufficient to enter and work through clinical trials. So we were really excited to see, and our clients have been really excited to see an actual guidance come from FDA that really spells out a lot of the key criteria for how to build your gene modified cell therapy, but also how to run a clinical trial associated with a gene modified cell therapy. So that guidance was quite helpful, and I think has definitely made it more straightforward in a lot of ways for our partners to enter the clinic. Of course, very recently, just about a year ago now, we saw the first approvals of a CRISPR-based therapeutic in Casgevy. Obviously, a very exciting moment for the field, and really speaks to the requirement in that I think Aldebaran is observing here, which is that clients are looking for scalable GMP solutions at this time. They're looking also for solutions that enable exploration of alternative deliveries of different CRISPR systems. So some of the ways that Aldebaran has adapted here, we launched in this period our mRNA sequence to vial manufacturing solution. So COVID, one of the, you could call it a silver lining, I suppose, was the proof at scale of the efficacy and broad safety of LNP encapsulated mRNA as a therapeutic vehicle. And so we've observed, huge explosion in companies and sponsors looking to bring into the clinic encapsulated formats of CRISPR drugs. And so at Aldebaran, we have the ability, all under the one Aldebaran roof, to go from mRNA sequence to delivery of encapsulated mRNA drug product. And so we leverage heavily our internal capabilities here, including the ability to make linear plasmid template and mRNA manufacturing enzymes. All of these things together make for a really robust solution for our clients who are seeking from a very early stage to take delivery of an encapsulated CRISPR-based therapy or CRISPR-based LNP. That's been exciting to watch the demand for that rise as CRISPR LNPs have been proven out as a potential safe, effective solution. We also, in this period of time, really doubled down on nanoplasmid technology. And you may be familiar with nanoplasmid, or at least the notion of an ultra small backbone plasmid template the original idea here, why Aldebaran brought this technology on, was to really help with AAV production. What we saw was that by using nanoplasmid, you really saw an increase in that transfection efficacy, and you saw a great decrease in double-stranded DNA toxicity when delivering nanoplasmid into your reaction. What we've come to find in working with partners is that this ultra small backbone plasmid also has a great application as a CRISPR HDR donor template. It turns out that this technology functions very well there. We've actually seen evidence from our clients that a benefit of using nanoplasmid is that you'll get a really nice recovery rate of modified cells after you go through your editing process. And so we've had several clients select nanoplasmid as their donor DNA source that they're going to put into their CRISPR reaction. So we've really been excited to see that take off as an application for nanoplasmid. So that brings us to today. Obviously, this is a ton of evolution in a very short period of time. You know, we've gone from, in about a decade span, from a totally new technology to approved drugs, a Nobel Prize, dozens more therapeutics coming through clinical development. And you know, as a field, I think we're poised to, you know, in the coming years here, treat many patients who were previously not treatable using CRISPR medicine. At the same time, I feel like we can recognize that there's a lot more work to do if we're really going to recognize as an industry the potential of genetic medicines, the potential of CRISPR-based therapeutics. In order to realize that, we need to reach many more patients. And on the CDMO side, we need to be thinking about how can we serve this market as we try to really expand the reach of these therapeutics. And so there's a few different ways that I'd like to explore with the rest of our time together that we see this market changing, we see the need for change, and I'd like to speak to those items. So on the supply chain side of things, presently, networks of suppliers can be quite complex for clients who are developing these drugs. That's one area where we see a need for some evolution. On the quality side, we've seen a shift, right? As previously we've been thinking in the clinical space, how do we get into clinical trials and through clinical trials? After approval by regulatory bodies, quality requirements typically heighten. Process validation, end-to-end manufacturing solutions are things that clients are working for. So those are challenges that we need to rise to be able to meet. And then finally, working in partnership with academia to come up with phase-appropriate manufacturing solutions that enable us to reach patients where they are in a cost-effective way that doesn't compromise on patient safety. So when we think about serving the clients in this space, typically what we see are that clients are working with a constellation of suppliers in order to manufacture clinical stage therapeutics. You're going to have a CDMO partner that you're working with for your CRISPR reagents, your nuclease or mRNA, your guide RNA, perhaps your HDR repair template. You may have a CDMO that manufactures your cell therapy for you. And all of these CDMO relationships are in addition to the internal work that you're doing to develop out your processes and determine your efficacy. So what we've heard from clients historically is that it can be quite challenging to navigate these networks of suppliers and align requirements across them. And so, you know, at Aldebaran, we alone can't serve the complete arc of the what's required in the CRISPR space. But when we look at Aldebaran in the context of our partner companies at Danaher, we start to realize that we have the ability to serve all the way across the development and manufacturing lifecycle of CRISPR therapeutics. And so a core focus for Aldebaran moving forward is partnership within Danaher to provide seamless solutions across these CRISPR spaces. One example of how we've accomplished this and are continuing to tighten up these relationships between these operating companies is the Aldebaran RNP manufacturing platform. So this is something that we've been offering to our clients for years. The idea is that rather than taking delivery of just a CRISPR nuclease, just a guide RNA at GMP, and having to work through that complexation event in real time during your clinical manufacturing, that Aldebaran could deliver to you in a single vial a pre-complexed RNP that has a suite of quality control assays wrapped around it, and is delivered in a ready-to-use format. So you have a consistent starting material every time you go to manufacture a dose of your modified medicine. Aldebaran, of course, does not make guide RNA, and so one of the ways that we've approached this is to work really closely with IDT to qualify in their materials and qualify them as a vendor such that clients can ultimately seamlessly enter into our manufacturing process. But also the vision here is to just with a single point of contact at either company, be able to purchase and take delivery of the RNP and then have Aldebaran just control and manage the process of actually taking delivery of the guide component. So reducing the burden of our clients to qualify on vendors, deal with vendor audits, and coordinate timing across two different CDMOs. I put this slide here. I would expect to see a lot more things like this in the future, where you see Aldebaran, IDT, other Danaher operating companies working together to provide true joint solutions to CRISPR manufacturing challenges. Finally, I wanted to highlight, as I mentioned earlier, there's a lot of patient populations for whom the current CRISPR boom is simply not reaching. And there's a lot of different reasons and motivations for that. But what we've had within Aldebaran, within Danaher, is a recognition that, in order to really broaden the reach of genetic medicines, that we'll need to come up with new ways to partner with industry, new ways to partner with academia. And so at Aldebaran, we're really excited to be part of what Danaher calls a beacon program that has been established with the Innovative Genomics Institute. And the idea of this program is that we're going to work together rapidly with IGI to deploy therapeutic solutions for a couple of ultra rare genetic diseases, familial HLH and Artemis deficient SCID. The idea here is that we're going to treat patients in a rapid turnaround using phase-appropriate materials that will enable clinical study and quick clinical manufacturing, but that the workflow here for how we're going to rapidly manufacture and deliver these reagents could be used as a template or a playbook, if you will, for other academic institutions who are interested in curing rare diseases, how they can also quickly deploy high quality CRISPR solutions that serve these patients and ultimately make a real impact faster. So early days with this beacon, where we'll be excited in future years to hear from the teams that are doing the clinical studies, how the therapeutic aspects of this have progressed. From our perspective, this is the first kind of program like this that we've been involved with a formal partnership. It's far from the last. So I would expect as time goes on, you're going to see Aldebaran, Danaher more involved in partnering directly with academia to help broaden the reach of these therapeutics. So it's something we're honored to be involved with, and we're really excited to see how it progresses here and make meaningful impact on some patients' lives. So with that, I really appreciate you taking this time, walking down memory lane with me. I hope this provokes some thought and discussion with you. And of course, if there's any questions or comments, please drop us a note. We'd love to hear from you.